We will move on to the methods of conservation of biodiversity. We know what are the threats for biodiversity loss. Now we will study about biodiversity conservation because when we have an idea how we conserve our biodiversity, we can when we get this awareness, we'll try to protect our this important resource called biodiversity, and we can thrive very well. Conservation is the protection, preservation, management, or restoration of wildlife and natural resources. There are two methods for conservation of biodiversity: in situ conservation, or it is called as on site conservation. The second method is ex situ conservation or off site conservation. The second method of conservation is ex situ conservation. Till now, we have seen the in situ conservation. That means whatever the plants and whatever the animals are present in their own habitats, they are conserved at their homes, and uh, they are maintained as and biodiversity is maintained thus. Here now the second type of method is ex situ conservation. That means taking these animals or any animal species or plant species from their native place or from their habitat, and they're moved on to another location, and they are protected there. This is called as ex situ conservation. Now, as we have told you, there are several methods for ex situ conservation like gene banks, botanical gardens, and genetic bank, gene seed banks, etc. So now we will see one after the other what ex situ conservation methods are. The tissue culture banks, long-term captive breeding, and cryopreservation, frozen embryo transfer is these are we will see one after the other. Like gene banks. The gene banks, these are the gene banks or seed banks. Gene banks is, you can also call as germplasm banks. In germplasm banks, what they will do is plant genetic resources of plants like pollen, the seeds, the branches, or the bulbs of the seeds, or tissues, and animal genetic materials such as sperms, oocysts, embryos, and somatic cells are preserved at ultra low temperatures, very low temperature, that is liquid nitrogen. They're stored in liquid nitrogen, that is at minus 196 degrees. So in these gene banks, they are stored. If they want any species to if there is any species lost, we can as well take the material from these gene banks that is stored here, and we can propagate it, and thus we can um, uh, can grow that species. So gene banks, they preserve the germplasm, and even the seed banks. We look at the seed banks. This is a seed bank. Here it is a seed bank, and a seed bank. Uh, the type of gene bank. It is also a type of gene banks where seeds of different crops and rare plant species are collected and stored for future use. Most of these seeds can be stored for centuries. They're chemically treated, and they can be stored even for centuries without damaging their genetic properties. Okay, now there is another method called as botanical gardens and zoos. Okay, there's botanical gardens and zoos, and there's animals in zoos. The animals from different habitats. For example, kangaroos from Australia, or rhinos from Assam, or Africa, or elephants from Africa, or any from part of uh, uh, the world. They'll be they will be imported, and they're kept in a place, and they are fed, and thus their progeny is protected, and they are protected, and their progeny is also protected. In the same manner, in botanical gardens also, rare plant species or plants which are endangered or which are facing a threat, they can be taken and they can be grown at a place, and they are protected. They are given nourishment. They are well looked after, and thus these plant species are protected in a ex-situ area. That means in another area they are brought from where they are brought. So this is the example for botanical gardens and zoos. Now, now tissue culture banks also. Here, tissues of different types of plants and even the animal tissues, they are stored and they are used for future use. This is another method of ex-situ conservation, is long-term captive breeding. Long-term captive breeding is the process of maintaining either plants or animals in controlled environments, such as here. Uh, what they will do is they will bring the plants or animals, and they will give a place to um, from them to um, from them to survive, and these species can be saved from extinction. Okay, and many deer species, turtle species, and many plants which are threatened species or about to extinct. These are best example uh, can be conserved through long-term captive breeding. This is cryopreservation or frozen embryo transfer, is as I have told you, and they are preserved. The genetic material is preserved under very low temperatures in the liquid nitrogen at -196 degrees centigrade, and thus they are preserved and they are used for future use. And this now we'll see the advantages of ex-situ preservation. The endangered plants and animals are in the through this ex-situ preservation method. Whatever the endangered plants or and animals are present, or whoever is whichever is verge of extinction, they can be successfully breeded at at a place, and they can be propagated and they're multiplied, and thus they can be protected. Even the threatened species are breeded in this captivity and then released into the natural habitats. And it is extremely useful for conducting research and scientific work on different species, because these ex situ preservation, all the controls are under our control. All the things are under our control. We can carry out research and scientific work on different species, and we can provide a very beautiful environment for them to thrive and to propagate and to live further.